they are there's so many, and they all work either alone or synergistically with others to create the diversity we observe in our ecosystems. Now, when we think of tropical forests, some of the ideas that come to mind is they have lots of trees, they have high diversity, they are usually quite large, the trees are quite large, and all of this. And so when we think of these tropical forests, we sometimes, especially when we often would forget that these trees are just like other living organisms, are competing for space, they're competing for other resources as well. And for them to be structured the way they are, it does look like there isn't so much going on, but actually there is quite a lot going on. So with tropical forests, they have very high diversity. They seem to have so much going on. And one of the things we try to understand is how do this diversity, so much diversity come to be with different species? How are they able to coexist? How is it that one species does not dominate? So these questions are some of the things we try to answer in regards to the composition and the structure of tropical forests. And for us to understand these ecosystems, we go to the basics. We try to understand what is happening below the canopy. And so we look at their dispersal, we look at the recruitment of seedlings, we look at, because this is where most of what is going on here happens. The structuring of tropical forests happens at this layer with the seedlings. And in describing recruitment or looking at plant recruitment, you are basically looking at how the abiotic environment, the biotic environment plays a role. And with plant recruitment, key processes that are important, we think of seed dispersal, we look at seedling germination, and how the traits of different species influence dispersal mechanisms, influence the ability of species to recruit, and also how the abiotic environment plays a key role in how these species grow and are maintained. Now, in my work, I do look at what is happening along this line: seed dispersal, seedling germination, and seedling growth with the biotic environment. So this is my focus, and this is what I will be discussing in this talk. Now let's take us back to what has happened in the last few years. The major event has been coronavirus. And if you remember, at the start of 2020, there was a lot of talk about this new virus and how it came from China, and people kept talking about it. And suddenly it was like every part of the world was infected. And if you remember that there was a lot of social distancing, and all of that kind of put the whole world in a sort of lockdown. And it was interesting to see on the news, I think in 2021, where a mathematician actually was, he looked at how much of this virus is there. And what he said was that all of the coronavirus all these virus in the world could fit into a Coke can, that 33 cl Coke can. And it wouldn't even, there would still be space in that can. Now, if you would agree with me, it is kind of baffling to think of how something so minute, if you look at the virus, they say a human that has the infection could have up to 10 billion of the variants in the body. Now, if you think of these viruses, they are so tiny, and yet they can have a major impact. And it is something that still baffles the world of science today, of how much these small organisms can create so much havoc and bring down a whole village in some places. And that was what we saw with COVID. And in tropical forests too, one of the major mechanisms that has been studied as driving tropical diversity and species composition is the conspecific negative density and distance dependence. And this mechanism is simply that when you are surrounded by neighbors of the same species, that's your conspecifics, you have a lowered chance of survival, or your performance is lowered. And this mechanism has been studied for a good number of years now. And it is said that, okay, with in these communities, if I have a neighbor that as a plant that is of the same species, we either would compete for the same resources, or we share the same enemies, insects, pathogens, and therefore the chances of, just like when we talked about social distancing, it's better for us to social distance ourselves to increase our survival. The same happens with plants. Now, one of the major hypotheses that's been put forward is the Jansen-Cornell hypothesis. And with the Jansen-Cornell hypothesis celebrated its 50 years, and it was celebrated the 50 years anniversary in 2021. And this mechanism has been, or this hypothesis has been around since Jansen and Cornell both worked on different research and showed that enemies, natural enemies of plants, are the key drivers of diversity in tropical forest ecosystems. Now, with the Jansen-Cornell effects, it is what it means is if you go into an ecosystem, if you go into a forest, trees disperse their seeds, and seeds would fall at high densities closer to the parents, and with distance, the seed shadow decreases. And what happens is, at this, be closer to conspecific parents, there are high attack rates of the natural enemies of these species. And therefore, the probability of a seed or a seedling surviving is higher the further it is away from its adult conspecific. So there are higher density and higher attack rates closer to the conspecific adult, but further away from this conspecific adult, there is a low density and there is also a low attack rate. Now, the Jansen-Cornell effect is said to have provide rare species an advantage, because with more common species, they would occur in more areas within the ecosystem, and therefore they have higher chances of meeting their conspecifics. And with rare species, the chances of meeting a fellow conspecifics are low. Conspecific is lower, and therefore this rare species have a higher chance of establishing within this system. And this is said to be what drives diversity, high diversity in tropical forests. And there has been many studies that have looked at this. There have been empirical studies, there have been theoretical studies that have looked at the importance of the Johnson-Connell effects in driving species diversity. And there are still many studies that are trying to understand how this mechanism plays a role in maintaining diversity in ecosystems. Now, I read about these studies, and many of these studies have been limited to neotropical the forests and Asian forests, and there have been very few studies in Africa. And so the question that came to mind is this mechanism, is it general? We have so many of these studies, and some of the conclusions, even from these neotropical forests and in Asian forests, are not quite in support of the Johnson-Connell effects. So I wanted to look at the role this mechanism plays in structuring forests in Africa, specifically montane forests. And for me, I thought, or I did think of the idea that it may be that the montane forests are the effects of the Johnson-Connell mechanism may not be as strong as what is observed in the neotropical forests, because montane forests have different features. They have less species diversity than your typical lowland tropical forest. They have lower temperatures, and therefore not quite suitable for microbes to thrive. And so I did think that, I should test this in a montane forest, and that is what I did. Now, tropical montane forests in Africa, the portions in green that you can see on the map, they are interesting ecosystems. They have fewer species than your lowland tropical forests. And the forests in the southern part of Africa, some of them are actually quite distinct from what you have in Eastern Africa and West Africa. Now, these areas are known to have high endemism and have unique diversity and unique organisms that are typical to just this environment. Now, previously with the testing of the Johnson-Connell hypothesis, one study was done in Ngel Nyaki by Arnie. Arnie was testing for the Johnson-Connell effects, and he carried out a study looking at the role of distance dependence on the distribution of several species. Now, in Arnie's work, he didn't make any. He did find some evidence of herbivory, and thus concluded of some conspecific distance effects. But for other species, there wasn't any conclusion, or there were no conclusive results of the presence of conspecific negative density dependence. And therefore, building on from Annie's work, I decided to look at, in a much larger scale, what the effects of conspecific density and distance is in this forest. So the question we asked, was, how do neighborhood densities affect seedling survival? and in Taraba State. It's a small forest area, as you can see on the Google map, surrounded by large grassland areas that are predominated by Fulani, and there's there's a lot of grazing activity that happens on the Mambila Plateau. The forest area, like I said, is quite small. We would classify this, I would say it's not a true tropical forest, because it does have a long dry season. So you have a 6 months wet and a 6 months dry season. It's a mountain forest, so high elevation. It has a good amount of rainfall, and the temperatures could go as low as 13 degrees Celsius, or even lower. But these are our means, calculated from over the years. Ngel Nyaki Forest has approximately around 123 species. Many of these species belong to single genera, which is quite unique. Now, in many tropical forests, you would find a genera that has, or you find one genus could have as many as 50 species. But in Ngel Nyaki, you find that most of the genera we have here are represented by only a single species. So there is a high genera diversity. The tallest trees in Ngel Nyaki are about 30 meters tall. And they all fall into different categories: evergreen, semi-deciduous, and deciduous species. Most of the species, like in many tropical forests, are animal dispersed. So 87% of the species here are animal dispersed. And just the photo on the right shows the kind of fruits that we collect from this forest. There are interesting animal species too, fauna, and these are just a few of them. There are very many bird species. As a lot of you, Joy, I can see Joy is around. Joy can tell you. And for people that have visited in Ngel Nyaki, there are other interesting mammals too. The chimpanzee is one that is often mentioned, which many do not see, but they are there, and they get caught quite often on camera traps. There have been studies on the African giant pouched rat and its importance in seed dispersal, the potty-nosed monkey, the tantalus monkey. They're all important fauna found in this ecosystem. Now, in 2013, which was my first time going to Ngel Nyaki, I went there primarily to start a project on the Ngel Nyaki forest dynamics plot. Now, this forest dynamics plot is a long-term plot that we established to study this ecosystem in detail. And this part of a network with the Forest Global Earth Observatory, and which is a network of about 77 plots, the last time I checked. And we all have the same methodology. So in these plots, we measured, we mapped, and identified all trees within a 20.28 hectare area. Now, the trees trees or saplings that had a size of 1 centimeter diameter and above were included in this census. So this gave us around 41,000 trees that we mapped and measured and had their IDs. Now, in 2017, I went a step further and decided to look at the younger life stage of these species, which we have, they're adults. And so I set up an additional 8,112 subquadrats where I had 1 meter by 1 meter tiny seedling plots. And these were spread around systematically around the Ngel Nyaki plots. And in them, I measured seedlings and saplings that were not greater than 1 centimeter in diameter. So I had 2 data that were useful in trying to understand the role of the Johnson-Connell effects in driving seedling survival. So this is to give you more detail. In addition to the 8,112 seedling plots, I also had this other seedling plots that I placed along with seed traps. So I wanted to understand seed dispersal, and I also wanted to see what was the relationship between seed dispersal and seedling recruitment. So all of this data, which amounted to around 11,000 seedlings, I had with my adult tree data, and I used it to carry out a neighborhood analysis. So I had a neighborhood model where I looked at seedling survival as affected by the identity of a seedling and also the density of a seedling and its adult neighbors. So I grouped these neighbors as conspecific seedling neighbors, heterospecific seedling neighbors, total combination of both. The model that looked at seedling survival as affected by these variables. And after my analysis, after I ran the model, this shows what variables were most important in determining seedling survival. So I also included height, because you would expect that a bigger seedlings would have a much more higher resistance than a smaller seedling would. So height was also included in our model. Now, on this figure, what you can see is on the x-axis is of a seedling surviving and how it is affected by these factors on the y-axis. So height, like we did, what we didn't find any conspecific negative density dependence. So conspecific negative density dependence was not a key factor in determining seedling survival in this community. However, total seedling density, so regardless of the identity of when there were more sounding an individual seedling, its chances of survival was low, closer or surrounded by more, their odds of surviving increased. Now, this was quite unusual for many of the tropical studies that that was out there. Because the usual finding is that conspecific adults have a negative impact on seedling survival. But in this case, we found that adult impact on seedling survival. Instead, adult important was useful in seedling surviving. But then this could be as a result of a lot of things. Many of these seedlings have different densities. So it could be that a more dominant species is driving this interaction. So we decided to look at different species, and because species vary to conspecific. So we looked at the growth form of the species that were in our community. So if these species were emergent trees, canopy, understory, or lianas. And so we looked at growth form as having an impact on seedling, on CNDD. And we also looked at habitat, where seedlings were growing. Seedlings growing at the forest edge versus seedlings growing at the core of the forest. Does edge effects play a role in a seedling's survival to neighbors? And then we also looked at shade tolerance. Species could be light loving. All species are light loving, but there are some species that are more sensitive to light compared to others. So we looked at how species vary in their response to conspecific neighbors. So in variation, we did find some variation across the growth forms. So if you look at the different growth forms here, you'd find that with emergent species, somehow similar effects to what we have with the general model. Total adult basal area is positive to seedling survival. With canopy species did reflect the common pattern observed in tropical forests. And so you would find with canopy species, conspecific densities played a significant role, and this is a negative role to seedling survival. So if you look at this figure here, you'll see that conspecific seedling density and conspecific adult basal area all had a negative role on the survival of individual seedlings. For understory species, it also did resemble what we had with the general model. And for lianas, quite similar as well. So the canopy species here make up most of the species that we recorded in our sampling. And these canopy species fall into different categories, and we could see that conspecific negative density dependence plays a very key role in the structuring of these species. When we also looked at the variation of conspecific negative density dependence across shade tolerance groups, we found that conspecific negative density dependence was a significant variable for light demanding species and intermediate light demanding species. So with light demanding species, you'd have the conspecific adult basal area was a key determinant of seedling survival and total seedling density as well. These showed that there was conspecific negative effects in determining seedling survival. But for the shade tolerant groups, there was nothing to show that. Now, just to kind of relate this to what others have found. In these systems, trade-offs happen, and you would find that due to these trade-offs, so with shade-tolerant species, they have traits that allow them to withstand attacks from enemies, and also because they already are living in harsher conditions. Like I said, all of these plants need light, but this shade-tolerant species, they're already in conditions where they have to adapt to the shade, and therefore they have traits that make them more resistant to attacks by natural enemies compared to light demanding species. So with shade-tolerant species, they are slow growers. Light demanding species are fast growers, and they are more susceptible to attacks by natural enemies compared to the shade tolerant species. So after these two field observations, because this that part of the work was based on field observations, I decided to pick out two of the mechanisms that are investigated as playing a key role in negative density dependence. So I decided to investigate negative soil feedback and also intraspecific competition. Now with feedbacks, the idea is that plants can change the biotic and abiotic properties of the soils in which they live. And these changes may in turn influence the growth of these plants. So a plant species may influence soil conditions in a way that could either support its growth or it will hinder its development. And so you could have where a plant modifies its soil biota in a way that would inhibit its conspecifics more than heterospecific neighbors, which we would call negative plant soil feedbacks. And you could have where a soil would modify its soil biota where it promotes the growth of its conspecifics, and then you call that positive plant soil feedback. So these plant soil feedbacks can shape the interaction among plants, and such as conspecific negative density dependence, facilitation, and competition. So I looked at looked at how plant soil feedback and intraspecific competition played a role in the survival and growth of 10 species in Ngel Nyaki forest. So I carried out shade house experiments where I used 6 of these species to test for plant soil feedbacks and 4 of these species to test for intraspecific competition. So with the plant soil feedback, what I did was I collected seeds from 6 species, and these 6 species, what I did was pick species from that were available at the time I was running this experiment, but also I was after species that were relatively common and those that were not so common. So I had different plants species selected, collected the seeds, and I grew or raised these seeds in the nursery for a few months. It differed because some species germinated really fast and others did not. So, but at 4 months, I had seedlings of these species. And then I collected from the adults of these species soil. So I collected soil from underneath the adults of this species, and this soil was going to serve as an inoculum for my experiment. So the assumption here is that the soil collected from beneath these adults would have the biota of associated with that species. And so I collected this soil from these 6 different, from the adults of these 6 different species. And when I transplanted my seedlings, I created an experiment where I had species that had soil from their conspecific adults and plants that had soil from the other adults. So I had conspecific and heterospecific setups. Then I also had a neutral or a control setup where the inoculum was sterilized. So this experiment had a 6-month duration where I allowed the plants to grow. Some of the plants died during the process, but at the end of 6 months, I harvested these plants and I estimated the biomass. For the second experiment, looking at intraspecific competition, I used 4 species, and what I did was grow these plants in high and low densities. So the assumption here, or the hypothesis here, is that higher densities there would be higher competition, and therefore plants would do poorly in these denser setups. And this experiment also ran for 6 months, and then I harvested and had the biomass estimated. Now, for the first experiment on plant soil feedbacks, my interest was in negative plant soil feedback. So I did look at how inoculum source affected the biomass at the end of the day, but we did not find any significant effects. There wasn't any significant difference between conspecific sources and heterospecific sources. And there wasn't any difference as well in the live and sterile inoculated plants. So we didn't find evidence for negative plant soil feedbacks in this experiment. And also with our intraspecific competition, we did not have evidence for intraspecific competition as well. Interestingly, what we found was that for this particular species, Tabana montana contota, it had a positive response to intraspecific competition. So with the log response ratio, anything above the 0 line shows intraspecific competition. So, but the intraspecific competition in these 2 species were not significant at all. However, this particular species where density seems to have a positive effect, that was quite interesting. And however, I wouldn't make any interpretations of this, because there has been a lot of debate about how important intraspecific competition is in driving patterns. And some researchers would say that with different species, you have to estimate how, at what density would you say there is competition happening. So what are the findings from this study? We did find support for negative density dependence. There was some evidence of positive density dependence too, which needs to be looked into. So do we have facilitative interactions? A lot of people would say the ecosystem at Ngel Nyaki is not harsh enough to have facilitative interactions, because facilitation occurs in areas like the in deserts and the alpine regions. But again, our evidence for positive density dependence could be as a result of strong dispersal limitation. Now, this is a plausible idea because with a study that we had done earlier, we found that most of the species in this area are strongly distal limited. So many species have very high limitations of dispersal. Many seeds fall around the parents and do not move away. So it's not that there isn't negative density dependence, but dispersal limitation is makes it look like most of the species that are present are closer to their adults. And so you would always find more of them around the adults. And this could be why we see some sort of positive density dependence. We also found support for conspecific negative density dependence in canopy species and light demanding species. And but no evidence for negative plant soil feedback and interest-specific competition. So these are like the summary of the findings from this study. Is more research needed? Yes, you would find the Johnson-Connell hypothesis has been around for 50 years, but more work is needed around this in this field. And this is because forests are changing, having more widespread anthropogenic influences, and this is having an impact on the species and interactions that shape this forest. I'll just draw your attention to the work of, to the paper that Redford wrote in 1992, where he talked about the title is The Empty Forest. And even though his work was about the loss of mammals in ecosystems, I think it is relevant for this particular study as well. Because sometimes we spend so much interest on the visible things that we see, the things we see with our naked eyes. In conservation, the focus is usually on the large mammals, the birds, the trees, and many times these organisms that are in the soil that play a significant role in structuring these forests are not investigated, they're not looked into. But we can see from so many of the studies that have been carried out that these interactions with this biotic environment in the soil plays a very significant role in the structure of this soil. Apart from the negative effects, there are positive effects from soil microbes. So when we study mycorrhizae play a significant role in the structuring of ecosystems. So I think some of the questions we should be asking is, can plant interactions be recovered? Can they be facilitated? And can they be initiated? So these are questions that I would like to leave for people that would want to venture into this kind of study. And yeah, thank you for listening. I'd like to acknowledge so many people who have helped me with this work, advisors, collaborators, the funding bodies, and especially my data collection team, Awal, Enoch, Dairu, Saliu, Helen, Joel, and all the others. Thank you so much for listening, and I can take questions if there are any.